So, hello everyone. Today we are going to start the second lecture of week 1, and today we will be discussing strain isolation and process flow. So, as we have already discussed in the bioprocess, you have a biological component which converts your substrate into product. Now, biological component may be an enzyme, it can be microbial cell, it can be plant cell or animal cell. So, today we are going to discuss particularly this microbial cells, and once we are going to use in the bioprocess, then first thing you have to isolate or identify that potential strain which can convert the substrate to your desired metabolite or product. So, we can quickly revisit what we discussed in the last lecture. We discussed what is the traditional definition of fermentation, particularly anaerobic fermentation. Then we discuss it can be anaerobic as well as the aerobic fermentation. Then what is bioprocess and what is the role of particularly bioreactor in the bioprocess development. Then we quickly revisited the history of bioprocess engineering, particularly from pre-19th century to 21st century, and then we briefly outlined different category of bioprocess products. Today we are going to discuss first isolation of potential strains, and followed by that we will be looking for strain improvement methodologies. So, followed by that we will see how you can improve the isolated strains to improve or to enhance particularly this yield of your desired product. So, first we need to have a biological agent. So, here we are discussing this microbial strain. So, as I told, the microorganisms are ubiquitous and they are found everywhere in the soil, air, water, food, sewage and on body surfaces. The role of microbiologist is to separate the mixed population into individual species, and then the culture containing a single unadulterated species of cell is called pure culture. So, one need to identify the pure culture. Typically in nature, microbial population do not segregate themselves by species, but exist as a mixture of many other cell type. So, whenever you are isolating from any source, you are going to have always the mixed population, and from mixed population you have to select a single species, and that is called pure culture. In laboratory, this population can be separated into pure culture. So, for example, these cultures contain only one type of organisms and are suitable for study of their morphological, biochemical and other properties. So, we need to have the most potential strain that can generate high yield of your desired product. Your desired product can be a biomass or your desired product can be metabolite. So, typically the suitable microbes are isolated from natural habitats, and further they are screened based on their metabolic capability, productivity, robustness and tolerance to the toxic metabolites and substrates. For example, if you want to select a bacterial strain which produces a high amount of biosurfactant, so one option is you can isolate from the site which is contaminated by oil, because for the degrading the oil there should be production of active compound biosurfactant. So, first you have isolated, then you have to screen. So, for screen you can screen based on the emulsification index, if it is forming good emulsion, either it is able to displace the hydrophobic ion molecule or it is able to break the layer of the ion molecule, that is a drop collapse test. So, based on these screening one can identify which one is the potential microbe, because once you are isolating you will isolate many, then you have to narrow down to the most effective microbes. So, the first thing you have to select the site from where you will get the desired strain. So, one approach is called the shotgun approach. So, it is a sample of free living microorganisms, biofilm and other microbial community are collected from animal and plant material, soil, sewage, water and waste streams, and particularly from unusual man-made and natural habitats. So, you are going to select from any location where you can collect the sample and isolate. Then these isolates are screened for desired traits, and this approach is useful for non-selectable traits, i.e., for example, antibiotic production. Another approach is objective. Objective means you are going to do the sampling from a specific site where it is expected that organisms with desired characteristics are present. For example, if you want to isolate oil degraders, you can select oil contaminated soils. Next approach is cultivation approach. So, here what you are going to do, you are going to have selective media. So, it says media and growth conditions are developed for obtaining the strain with the desired traits, and in this case screening relies on media staining or individual testing. And the last approach is you can use any convenient source. These are public culture collections, for example, ATCC, American Type Culture Collection USA, or MTCC, Microbial Type Culture Collection Chandigarh, and there are many more. Once you have collected the sample, then you are going to apply these pure culture techniques. So, it basically it is a streak plate, pour plate and spread plate, and for this you require certain facilities, like you require media, that should be a solid media as well as the liquid media. You will require the autoclave, you will be required different utensils like culture tube, petri disk, pipettes, and further for you require cultivation or incubation chamber, and of course you require refrigerator to preserve them. So, these are basic laboratory facility for the isolation of microbes. So, you might have discussed in detail in your microbiology course, but here we are going to briefly discuss these pure culture techniques, particularly this streak plate, pour plate and spread plate. So, you can see here what we have discussed, you have to isolate a strain. So, for that you have to collect the sample, and I had told there are 4 different approaches for the isolation: shotgun approach, objective approach, cultivation and convenient source of microorganisms. Then you can have different isolation techniques: streak plate method, pour plate method, spread plate method and serial dilution methods, and from there you are going to identify the selected strain. Then you have to identify and characterize. So, for this you can use microscopic examination, different biochemical assay, molecular techniques and metabolic profiling. So, these things we will be discussing in subsequent slides. So, you can see pour plate method, you are having an agar plate and you are pouring the solution which is having your sample here. Here you are spreading on the petri disk having solidified agar using a bent glass rod. In the C you are streaking. So, this is a pouring, spreading and streaking. Now, next one you are going for the serial dilution. You can have different dilutions, and as you are increasing the dilution, you can see the number of colony are significantly decreasing, and what is assumed that one colony is formed from individual microbe. So, based on this we also calculate what is the concentration of microbe in terms of colony forming unit per ml, CFU per ml. It says number of colony into dilution factor divided by volume of culture. So, what was the amount of culture you added for the dilution. So, here you can just see I have these are just representative. For example, on agar plate you can see different morphology like circular, irregular or branched structure. Even you can see this linear structure and some convex structure as well. In addition, you can see filamentous structure or some other different kind of structures. So, based on different morphology we identify these are different population of microbes. Other approach can be you can go for this enzymatic assay. If let us say gelatin liquefaction. So, if it is microbe is able to produce gelatinase enzyme, it will be able to liquefy this gelatin. Other option is you can grow in the broth, and by growing you can see different turbidity as well as different sedimentation rate depending on the microbe. So, we can see these are the likely to be different populations. Further, in the case of bacteria, one can go for the gram staining, whether it is a gram positive or gram negative. So, gram positive have a thick layer of peptidoglycan as compared to gram negative bacteria. So, here what you have to do, the first step is application of crystal violet, then followed by the application of grams iodine, then you are going to wash with 95% alcohol, and then finally you will see this, you will add this safranin. So, gram positive cells are purple, while gram negative bacteria which have attached the safranin, they becomes in the pink color. So, here in fact when you are washing with the alcohol, gram positive are still purple while gram negative becomes colorless. So, what happened? Gram negative are having thin layer of peptidoglycan. So, they are washed by the alcohol while the cell wall is still intact for the gram positive cell. That is why the gram positive is still purple here, because they did not retain safranin, while the gram negative they retain the safranin. So, next, what is the biochemical test? The microbiologist benefit from the fact that similar to humans having unique fingerprints, bacteria also contain distinct biochemical identifier, and these so called biochemical fingerprints are properties controlled by cells enzymatic activity. because enzymes are very much specific and they are responsible for bioenergetics, biosynthesis and biodegradation. And some of these chemical reactions are called cellular metabolism and biochemical transformation that occurs both outside and inside the cell are governed by these biological catalysts or enzymes. So, we can have this particularly different enzymatic assay. Let us say you can have this starch hydrolysis, amylase, lipase or proteinase assay. So, one option is you can have the activity of extracellular enzymes, you can have the activity of intracellular enzyme. In addition, there are certain tests, that is the IMVC test. I for indole, it detects the ability to break down tryptophan to indole. Second is M for methyl red. M for methyl red, this checks the stable acid production from glucose fermentation. V is for bogus Proskauer test. So, V, what it does, it detects the production of acetoin, which is a natural end product of glucose fermentation, and C is citrate utilization test. This determines if the organism can use citrate as sole carbon source. In addition, there is another test, that is the triple sugar iron test. So, triple sugar iron test is a biochemical test used to differentiate gram negative enteric bacteria based on their ability to ferment sugar and produce H2S, and this H2S is particularly detected by this iron solution. So, you can see you can have the biochemical test based on the extracellular or intracellular enzymes. Other option is you can just see this, they are based on their particular metabolic pathway, intermediates or end product. Next, one can also go for metabolic profile, which identifies and quantifies the metabolites of a novel strain. This helps to reveal the strain's unique metabolic capability. In fact, it enables the understanding and classifying the biochemical pathway which a particular microbe is going to follow. It supports the optimization of growth conditions and it identifies the key metabolites for biosurfactant production and other industrial uses. In fact, it enables to identify the key metabolites. For example, you can see based on the metabolic profile whether they are going to produce biosurfactant or any other metabolites. So, you can see you started with strain isolation and cultivation and prepared your sample, then extraction of intracellular and extracellular metabolites you can characterize using GCMS, LCMS or NMR. Then here it goes this metabolic pathway analysis. So, what we are going to do, identification of key metabolic pathway, even you can use database of like KEGS, Metasys or HMDM. So, KEGS is quite common, this is the Kyoto Encyclopedia of Genes and Genomes, and HMDM is Human Metabolome Database. So, there are different database which enables to see the pathway, and experimentally you have already identified few metabolites, and these pathway are specific for a microbe. So, there you can see a microbe can follow the different pathway and your intermediates are coming into the in which particular pathway. So, based on that you can decide which pathway is being used to produce a particular product from a given substrate. Next goes this molecular techniques. So, this genomic technology are replacing the traditional microbial classification methods. So, it focuses more on the gene sequence and cellular and molecular component. And in fact, if you want to give a particular name to an isolate, then you have to go for these molecular techniques. For example, this taxonomic identification and understanding their metabolic profile. So, for this there are common techniques like 16S RNA, whole genome sequencing, metagenomic analysis, so on and so forth. So, in this way one first identify this genetic sequence and based on that we identify the microbial strains. So, once you got this sequence using bioinformatics tool, then you have to use this blast the sequence into gene bank to obtain a match. So, it gives whether this is a 99% similarity, 90% similarity or it is less similar to the a particular species. So, once you have isolated, now next comes you have to go for the strain improvement. What it says, after screening the selected species are subjected to strain improvement. Why we need to go for strain improvement? Particularly to improve the yield of your desired product. So, this enables manipulating and improving the microbial strain to enhance their metabolic capacities for production of valuable products. So, for this different kind of genetic and metabolic engineering tools are applied to improve the performance of wild species, which is particularly identified from the natural source with respect to type and quantity of product. In fact, it is possible to redirect the carbon flux in such a way that productivity or yield of a desired product is improved, or you can introduce the new metabolic pathway in suitable hosts. So, what it says, a metabolic pathway or substrate is converted to one product and different byproducts. It is definitely possible that you can stop those other byproducts, so that all the carbon flux will go for your main product. And in fact, with the metabolic engineering and genetic engineering, it is possible that you can use any host to produce any novel metabolites by applying the genetic engineering. So, for these few approaches I will discuss here quickly. One is the random mutation. You can apply some extreme environment like X-ray, UV and other chemical mutagens, so that it is going to apply some stress on microbe, and under stress they will try to develop some different phenotypes. So, for example, penicillin yield has been significantly improved by using random mutation to the strain Penicillium chrysogenum, which is now commercially used for the penicillin production. Other option is heterologous protein production. In this case, a heterologous gene is inserted into the production host to initiate a de novo protein synthesis process, and it has been applied for the production of various pharmaceutical proteins like hormones, antibiotics and vaccines and novel enzymes. So, you can insert a heterologous gene. Other option is protoplast fusion. You combine the genetic material of two different species in a single cell, and it has been successfully used in the yeasts and fungi. Next approach is transgenic techniques. Here genes are transferred from one organism into another and are transcripted in the recipient organisms like transgenic plant, transgenic animals, and here comes this metabolic engineering or pathway engineering. So, new metabolic pathway are introduced in the host organism to improve the substrate utilization efficiency, to eliminate the byproduct formation or to synthesize a new product altogether. So, when we are doing this strain improvement, typically we go with this generally regarded as safe microorganisms, grass microorganisms, because they do not require further this ethical approval. So, I have listed a few species of bacteria like Bacillus subtilis, Lactobacillus bulgaricus, Lactococcus lactis, and in the east the Candida utilis, Saccharomyces cerevisiae, and filamentous fungi Aspergillus niger, Aspergillus oryzae. So, these are the strain which are most commonly exploited through metabolic and genetic engineering. So, here you can see you are having a host microbe and different biochemical assay you got the gene of your interest. Now, you have to add that gene of that interest into the plasmid. So, you form a recombinant plasmid. Now, you insert this recombinant plasmid into the gene, then it has come to the host. Now, this it is dividing and you have tested this gene expression. Now, once you got this engineered strain or engineered microbe, then you are going for a small scale culture, then benchtop bioreactor 1 to 5 liter, pilot scale reactor maybe 100 to 1000 liter, then you are going for industrial scale operation. Here the scale will be few thousand liter, and go for product recovery and finally packaging and marketing. So, few example which have applied this recombinant technology and produced insulin, growth hormones like erythropoietin and interferon gamma. So, I will stop here this lecture. So, today we discuss different approach for isolation of strain as well as its improvement. Thank you.